Thank you to the organizers for having me. Nice to see so many friends and partners of Ori in the room. Thanks for coming. For those of you who I don't know, my name is Jason Foster. I'm the CEO of Ori Biotech. Ori is a company that's on a mission to enable widespread patient access to life-saving cell and gene therapies. It's always great to take a moment to just reflect on the successes. We spend a lot of time during conferences like these talking about the challenges. I was fortunate enough to meet a little boy called Opie Jones. Opie was 1 year old when he was diagnosed with ALL. His parents, Lucy and Lewis, took him to Great Ormond Street. He went through the normal protocols that you would imagine, the standard of care. Unfortunately, relapsed after a bone marrow transplant and was only then eligible for CAR T therapy. Opie responded very well and is now 2 years cancer-free from his leukemia. Lucy and Lewis have gone on to found the Opie Jones Foundation, which is really meant to support the families of those going through CAR T therapy. And I heard a story from Lucy that Opie was only one of 9 children of his age group that was eligible to receive CAR T therapy in all of Europe. You think about Europe, a population of 540 million people, 50% larger than the population of the US. Clearly, access remains a challenge for us. If you look globally at access to CAR T therapy, we're treating less than 3% of the available patient population. That represents about 21,000 patients so far over the last 5 years. If you look across the 6 approved CAR T therapies, together they treated in 2022 less than 7,000 patients. Now, it's a great milestone, up about 50% over the prior year. It looks like we're again set to grow that patient-treated population by another 50% in 2023, but it's still only a small drop in the bucket for the number of patients that could potentially benefit from these life-changing therapies. I've also had the pleasure of meeting Amy Kappen and her family. Amy's daughter, Sophia, similar story, leukemia. She was diagnosed at a young age. I think she was 4 years old. Within a year, unfortunately, after going through the standard of care and even receiving CAR T therapy late in her treatment, Sophia passed away. We know that the reality is today that, unfortunately, many patients are not getting access at all to these therapies, and the ones that are getting access too late, fourth-line therapy when these products show clinical efficacy at first and second line. Why is that? We spend a lot of time talking about these challenges. I think the realities are today's cell and gene therapies are too hard to manufacture. We just can't make enough of them. We can't make them at a high enough quality, low enough cost to make them widely available. Peter Marks made a comment recently publicly that said that for CAR T therapies to get increased access, the prices have to come down dramatically. And to do that, we have to drop cost of goods. We have to give manufacturers pricing flexibility so they can contract in creative ways with payers to really make these products available. Right now, we are developing products and we are launching products into the market that are not commercially viable, and they're never going to be able to treat large volumes of patients. And that is not what any of us in this room spend our days and nights thinking about. That's not why we are in this industry. So Ori's approach combines these 3 elements. Novel biology, so it's a novel bioreactor system, bringing engineering and automation and digital together as one to solve both the flexibility challenge for early stage development, but also the scalability challenge for late stage manufacturing. Over the last several years, we've been focused on biology first. Cell and gene therapy starts with biology. We start with cells. Can we get the right number of cells, the right phenotypes, the right viabilities? Do they have potent tumor-killing capability? And we've spent a lot of the last few years demonstrating that we can get the right numbers, the right phenotypes, the right viabilities out of the system. We've done over 500 runs in the system over the last couple of years with partners both in North America and in Europe and internally to really understand and characterize this new system. We've done runs using over 30 donors and patients. We know that there's a lot of donor-to-donor, patient-to-patient variability we're trying to account for in our processes. We wanted to be able to demonstrate the repeatability and the reliability of the process. We've done over 10 unique processes using cell types like CAR T, TIL, TCR, dendritic cells, CD34s, really running the whole gamut of suspension culture. Again, demonstrating the flexibility of the platform. From the very inception of Ori, We've been trying to create a platform that's flexible enough to flex to the needs of your process versus you having to shoehorn your therapy process into the constraints of the existing technology, which is really today's standard. We worked with 9 partners, 5 therapy developers, a couple world-leading CDMOs, and MD Anderson Cancer Center down in Houston, where our technology is currently deployed at the CTMC. Jason Bach may be in the room somewhere. We've demonstrated the ability of the platform to do a range of unit operations. We're really focused on the longest part of the process, the most difficult. Activation, transduction, expansion represents about 70% of today's cell therapy manufacturing process. We can go from a very low volume, maybe 30 mil and 30 million cells, up to 1 liter, and to get a cell yield of over 12 billion cells out the other end. So really demonstrating the flexibility of the platform and the ability to deliver on your quality, the critical quality attributes that you're trying to deliver for your product. So now a bit of data that this set of data and set of experiments we've generated internally. So this is our in-house process using Transact as an activation reagent. This was 6 different donors over 24 different runs to really demonstrate the robustness of the platform using the same exact protocol in both the ORI system and a control. The control in this case being the leading cell expansion system used across many processes, using the same raw materials, the same cellular inputs, the same reagents, et cetera, as best as we can do to get an apples-to-apples comparison. Here we're just trying to optimize for cell yield in this set of experiments. So a standard process, by day 6 or 7, we start to see statistically significant separation from control on a cell yield, cell count measurement. By day 10, we're seeing something like 2.5x the number of cells yielded out of the system in that kind of 10 to 12 billion range consistently. And this is starting with 70 million cells, so you're looking at 170 to 200x fold expansion. The question I often get after that graph is, well, aren't the cells exhausted after all that expansion? The good news is using standard markers like PD1, TIM3, LAG3, we're not seeing any signs of exhaustion. We're not seeing any differences in CD4 to CD8 ratio from both the starting material and also from the control, and we're not seeing any differentiation caused by the system. So we want to make sure that the system itself isn't causing the cells to differentiate. Looking at another set of experiments, this time instead of using GFP lenti, which we were using before, we're using an off-the-shelf CD19 CAR. This is a second-generation CAR, really looking at transduction efficiency and transduced cell yields rather than just total number of cell yield. So by day 7, in these set of experiments, we were using 4 runs with 2 donors. We again started to see statistically significant separation from control. Day 7, looking at around 2 billion transduced cells in the ORI system, 6 billion total cells, and then by day 9, about 4 billion transduced cells with close to 10 billion total cells in the system. The question also often comes up then, well, are these cells potent? Do they have cancer-killing capability? So using industry standard killing assay or cytotoxicity assay, NALM6, the cell line co-culturing alongside these cells, and also the CD19 negative cells, cell line K562, we show essentially specific killing for the CD19 positive cells, but not indiscriminate killing with the CD19 negative cells. That's what you would expect to see with this kind of assay. On the cytokine release assay, using again standard measurements like granzyme B, interferon gamma, and TNF alpha, we're again seeing potency and cell killing activity for the cells coming out of the system. Now, most of you will be familiar when often people show data from their own labs, it looks great. When it gets out in the wild, it looks a little bit different. We've deployed technology with 4 partners. This is data from one of those partners using their own in-house CD19 CAR with their own success criteria, 3 runs with 3 donors. The ORI system outperformed control in all 3 instances, in this case, with a highly variable distribution. So the system yielded 3.5 billion cells in one, 2.5 in the other, and then below 1 billion. I think this is indicative of the kind of donor-to-donor or patient-to-patient variability we see. And you have the opportunity, you can see with the run 3, which was the lowest of them, we had a slow-growing donor. But the idea that potentially if we were to extrapolate that out to day 10, we could rescue that that dose for that patient if it were a patient therapy being manufactured. We were able to hit the fold expansion target, the viability target, as well as the transduction efficiency target. In this case, they were using an MOI of 1.5. The number of transduced cells was about comparable across the two systems. In this case, we were a little bit ahead in the second one. The third one, unfortunately, the control failed in the process. Similar results across the phenotypic assays, showing no real differences between the ORI system and control. And then in their in-house potency assays showing cell killing potential. So that's looking backwards. Now let's look at today and what's happening in the future. So we're currently working with 5 Lightspeed early access partners, so LEAP partners as we call them, to really optimize the system more as we move forward. So we want to optimize things like transduction efficiency. Can we use less virus to cut cost of goods? Can we get just as many transduced cells out? Can we activate in a more optimal way? Can we shorten process times? So the data that I've just shown you is really trying to optimize for cell yield. What if we could get to, I'm half Italian, so I talk with my hands, apologies. We're trying to optimize for time to a certain cell number. So, trying to get to 2 billion cells or 4 billion cells, whatever the target is. But could we do that we think in maybe 3 to 5 days instead of 7 to 10 days? That has really positive knock-on effects. Looking at reducing tech transfer times. With partners, generally we're doing tech transfer within a month. And we're then starting to optimize their process beyond with that they've been working on with their current tools. We're building advanced analytical models so you can actually do some of this work in silico rather than going to the lab and doing wet experiments, which we know are expensive and fraught with errors. This is what's happening now, and this is what we're up to in the system or in our development today. So looking to move beyond biology, we've proven without a doubt that we can get the biology out the other side of the system, but we can't scale the biology today. That's the big problem. We can get the biology out of a bag or a flask, but we can't scale that biology. We can't make it cost effective. We can't make it widely available. So it really relies on these other components, the engineering and the digital components, to get us from R&D into the clinic quickly. If we can shorten tech transfer times, if we can actually develop on a platform that's flexible enough, we can move seamlessly from R&D into the clinic and from clinical scale to commercial scale. Recently, FDA put out some guidance that said, essentially, we recommend that you don't make any manufacturing changes after you hit the clinic. I think we all know that that's not the way we work today, but if we have platforms that are both flexible and scalable, we can actually think about doing that. So the ORI system is flexible enough to do early development, supporting all different culture types, active culture, static culture, large volume, small volume. We've automated fluid handling, so you reduce touch time, you reduce operator requirements. You can automate and get homogeneous sampling out of the system. You can do it with 1, you can do it with 3, you can do it with 7, you can do it with 10. It doesn't matter what scale you want to get to, you can get there using our system. If you can develop your process on the ORI system, then you can ultimately know that you can guarantee yourself commercial scale manufacturing. With second layer automation, we have the ability to then service the devices in a multiplexed way, dramatically shrinking the footprint that you need to deliver thousands of doses a year to patients. All that comparability risk, all that translation risk that we suffer today as we change our process at each phase is eliminated. The other thing is a completely IoT cloud-native system. So you can see into the cell culture in real time. You can capture the data you need, transfer it to an e-batch record system, do things like continuous validation, and do things like release by exception. Ultimately, we can create a high-throughput manufacturing platform, but if we can't batch release in the same fashion, we're stuffed. You've got a 1,000-page paper batch record, you're ticking boxes off one at a time, it's going to be hard-pressed to get to scale. These innovations have tangible benefits for our partners. So there's cost savings, there's quality benefits, and there's also time savings. These are indicative. Every process is unique. Every process is different. We dramatically reduce the labor required to manufacture a single dose or develop a product. We dramatically reduce the size of the footprint of the facility you need to make these products. We reduce cost of goods. We reduce tech transfer time. We likely can shave 20 to 30% off your drug development timeline while improving quality, increasing throughput. I think we can all agree that the current status quo isn't good enough. It's not good enough for patients. It's not good enough for the industry. We have an incredibly promising modality that really isn't reaching its clinical potential because we can't treat patients with it. So we urgently need to solve for widespread patient access. If you want to learn more, you're invited to come visit the team, come visit our labs. We've got a couple members of the team here, so Kale, put your hand up if you don't mind. Kevin and Matt on the digital team. Tom's probably here somewhere. Come talk to one of us. We'd be very happy to host you in our labs in Princeton or in London. Come see the technology. We can demo it. We can transfer your process onto it. We can demonstrate that we can deliver your protocol and your process on our system. And we look forward to enabling widespread patient access to these therapies with you as a partner. Let's go manufacture brighter futures together. Thank you.